This video is for Paper 1 of GCSE Biology and Combined Science. You can get extra help with this exam by downloading my revision workbooks and mind maps from emmatheteachy.com. The link is in the description below. I really hope you find this video useful. If you do, you can support me by giving me a thumbs up and subscribing. GCSEs are tough, but the fact that you're watching this means that you care and are putting effort in, and that always pays off. Good luck for your exams, and thanks again for watching.
Our first topic is cell biology, and we're starting with number 1, microscopy. We're going to look at two types of microscopes, as you need to understand how microscopy has developed and the differences between these two. First up is a light microscope, which you'll have seen and used in school. It was first developed in the mid-17th century, and it uses light to form an image. Because of this, it can be used to view live specimens, for example, bugs. It's also pretty cheap, and it's easy to use, which is a big advantage. The best ones can magnify up to 2,000 times, which means that it zooms in 2,000 times. Resolution is all about the details you can see. It is the ability to see two things as separate objects. A high resolution means you can see lots of detail and everything is clear and separated. The light microscope has quite a low resolution, with a resolving power of around 200 nanometers. Now let's look at the electron microscope. It was developed by scientists in the 1930s, and it uses electrons to form an image. It does this by firing electrons through the specimen, which means that the specimens must be dead first. So it can't be used to view living specimens. It's a very expensive microscope, and it has a lot of conditions that it needs to be kept under. However, the rewards are really good, as the magnification can be up to 2 million times. And it has a really high resolution of just 0.2 nanometers. The magnification and the resolution are the two big ones you need to remember to be able to compare these microscopes. In both cases, the electron microscope is much better. It has really high magnification and resolution. And this has allowed scientists to see and understand lots more about the subcellular structures of cells. We'll look at these in more detail in the next video. Number 2, animal and plant cells. We're going to start with animal cells, and we'll look at the organelles that it contains and the functions of each. So the first organelle is called the nucleus. That's this big black blob. It controls the cell's activities, so basically everything. And it also contains genetic material, which in animal cells is DNA. On the outside of the cell is the cell membrane. This is responsible for controlling the passage of substances in and out of the cell. So that's things like glucose and water. Down here, we've got the cytoplasm. This is a liquid gel that fills up the entire cell, and it's where chemical reactions occur. So make sure you learn chemical reactions happen there. This squiggly guy is a mitochondrion. That's the singular. And if you had a few of them, we'd call them mitochondria. That's the plural. Okay, their function is super important. They are where aerobic respiration occurs, and that releases energy for the cell. So they're really important. Now we've got a little tiny black dot over here, and we call this little guy a ribosome. You can also sometimes see the ribosomes attached to this structure, and you'll learn more about this if you do A-level biology, but for now, just look for the little tiny black dots. Its function is protein synthesis, which is just a fancy way of saying making proteins. So these are all the parts that you need to learn for an animal cell. Now let's look at a plant cell. I want you to pause the video and see which parts you can already label. You'll know some from what we've just covered. Ready? Okay, so the big black blob is the nucleus. That's the same in the animal cell. And we've also got the little tiny black blobs, which are the ribosomes. Over here, we have got the squiggly guy, which is a mitochondrion. And we've also got our liquid gel, which is the cytoplasm. Okay, we have got the cell membrane, and that is the inner layer of the two layers that you can see. And I'll go over how you remember that a little bit later. So now let's swap colours and we'll do the parts that are specific to plant cells. Starting with this green blob, this is a chloroplast. Now, not every plant cell has these, but when they do have it, their function is to contain chlorophyll, which is that green pigment that makes it look that way. And chlorophyll is really important because it absorbs light for photosynthesis. You're going to do a lot more on photosynthesis later on in paper 1. We've also got this big white blob, which is a permanent vacuole. Its function is to be filled with cell sap, and that's important because it helps keep the cell rigid. You'll notice that plants don't have skeletons, so they need this to help support them. And down here, we've got the outer layer, which is the cell wall. And my trick to remembering this is that the wall is always the outside layer of your house. And it's the same in a plant cell. It's on the outside. In plants, it's made of cellulose, but in fungi, it's made of a different material. Okay, and just like the wall of your house, it strengthens it and gives it support. So it's really vital. Also vital is that you learn the differences between plant and animal cells. 3. Eukaryotic and prokaryotic cells. Animal and plant cells are both eukaryotic cells, as well as fungi and protista. We call them eukaryotic cells because they all have some features in common. They all have a cell membrane, a cytoplasm, and genetic material that is found inside a nucleus. Prokaryotic cells are different. All bacteria are prokaryotes, so we're going to look at a bacterial cell to see the organelles that it contains. So they've still got a cell membrane, and they still have a cytoplasm, and they've still got ribosomes. They always have a cell wall, unlike eukaryotes, which only sometimes have a cell wall. And unlike plant cells, it is not made of cellulose. The next big difference is that their genetic material is not enclosed in a nucleus. Instead, it's found as a single loop of DNA inside the cytoplasm. There may also be one or more small rings of extra DNA, which are called plasmids. And some bacterial cells may also have a slime layer, which is for protection, and they may have a flagellum, or plural flagella, for movement. It's important to learn the differences between eukaryotic and prokaryotic cells. Notice that prokaryotic cells also don't have any chloroplasts or mitochondria, as they're just so small that these can't fit inside them. 4. Specialized cells. When a new organism is growing, initially all of the cells are the same, i.e., they are undifferentiated. As the organism develops, the cells do differentiate. This means that they change by developing different subcellular structures. We'll look at examples of these in just a moment. We call these cells that have developed the subcellular structures specialized cells, as they are able to carry out a particular function, or that's just a fancy word for a job. This will be in the tissue, organ, or whole organism in which they are found. We're going to look at 3 specialized animal cells. You need to be able to explain how the structure of a cell relates to its function, so we'll cover this for each cell, and you can expect to be given some information to use for these types of questions. So first up, we've got a sperm cell, and its function is to reach and fertilize an egg cell. Can you spot any structures that would help it do this? Well, first up, we've got a tail that helps it swim or move through the reproductive system to the egg. Then its midpiece has got many mitochondria to release energy for the movement of the tail. Up here in the head, we've got what we call an acrosome, which contains digestive enzymes to break down the egg and allow the sperm cell to penetrate it. And of course, we need a large nucleus to contain DNA, which can then be passed on to create offspring. Down below, we've got some muscle cells. Their function is to contract and relax, causing movement. Can you spot anything that would help them do this? Well, over here you can see that we've got lots of tiny little pink things, which are mitochondria. These release the energy needed for the contractions. Over here we've also got some special proteins. These cause contractions by sliding across each other. And finally, these cells can also store glycogen. This is broken down into glucose to allow respiration to happen. You're going to learn more about respiration in the bioenergetics topic, so don't worry too much about that now. Finally, up here we've got a nerve cell. Its function is to carry electrical impulses around the body. What can you spot that would help it do this? Well, this is called an axon, and it's very long to allow the impulse to be carried along it. And over here, you can see we've got lots of these little spiky things, which are called dendrites. Dendrites allow the nerve cell to connect to lots of other nerve cells. And finally, we've got nerve endings. These release chemical messengers that will cause an electrical impulse to be carried in the next nerve cell. So the nerve endings have lots of mitochondria to release the energy needed to make these chemical messengers. Now we've got our specialized plant cells. We're starting with the root hair cell, and its function is the absorption of water and mineral ions from the soil. What can you spot that would help it carry out this job? Well, first of all, this little protrusion provides a large surface area for the greater absorption of the water and mineral ions. We've also got a large permanent vacuole. This speeds up osmosis, which overall will bring more water into the cell. And again, we've got lots of these guys, many mitochondria. These are going to be for the active transport of mineral ions, and you'll learn a lot more about this in the active transport part of this topic later on. Next up, we've got the xylem. These cells are responsible for the movement of water and mineral ions up from the roots through the rest of the plant. What can you see that would help them do this? Well, you can see that they form long hollow tubes, so the cell walls have been removed, which allows the easy movement of water and mineral ions through them. They also have spirals of lignin. This allows it to strengthen the cells to withstand the pressure of the water moving through it, and it also supports the plant as it doesn't have a skeleton. Our sixth and final specialized cell is the phloem cell. What can you spot that would help it move dissolved food up and down the plant? Well, first of all, you can see that the cell wall has been replaced by these funny little things called sieve plates. And just like a sieve, they've got holes in them to allow dissolved food to move easily up and down through them. We've got another type of cell here, which is called a companion cell, and it's adjacent to the phloem cells. These have lots of mitochondria, and they release the energy that allows the dissolved food to be moved up and down through the plant. 5. Diffusion. Let's start with a definition. Diffusion is the spreading out of particles of any substance in solution, i.e. a liquid, or a gas, resulting in a net movement, which just means overall movement, from an area of higher concentration to an area of lower concentration. Let's look at an example of diffusion. So here we've got a beaker of water. Water is a liquid, so its particles can move freely around. Now let's imagine that you drop some food colouring into it, like this. The food colouring is really concentrated in one place initially, so this area is a higher concentration. Everywhere else in the beaker is a lower concentration, since there's no food colouring there yet. So we can label it like this. Because it is in solution, the food colouring particles can also move around. The movement of these particles is random, and they spread out from a higher concentration to a lower concentration. This can also be described as down a concentration gradient, as they're moving from high to low. So we can show it like this. Eventually, all of the food colouring has completely diffused throughout the beaker, so that the concentration is equal everywhere. If you were looking at this, it would look like a pale purple colour. The particles will still continue to move around randomly. It's worth noting that diffusion is called a passive process, and this is because no additional energy is needed to move the particles down a concentration gradient. There are three factors that can affect the rate of diffusion. The first is temperature. Higher temperatures cause particles to move more quickly, so diffusion will happen faster as they spread out much more quickly. Using a Bunsen burner here would make the food colouring diffuse much more quickly. Similarly, if there were a cold temperature, this would slow down the rate of diffusion. The second factor affecting the rate of diffusion is the concentration gradient. This is the difference in concentration. The greater the difference in concentration, the faster diffusion will occur. Look at these two boxes. In which one will diffusion of the orange gas happen the fastest? Box A or box B? Pause and see if you can work it out. Ready? Well, we can see in both boxes that the high concentration is up here in the top left corner, so we can label it as such, and then we'll need to compare the rest of the area of gases. So here we have got a lower concentration than the top left corner, but in B it is even lower as there are no orange gas molecules, and therefore it's a steeper concentration gradient, so diffusion will be much faster. It will still happen in box A, but the diffusion will be a lot slower. Okay, finally, our third factor affecting the rate of diffusion is surface area. Having a larger surface area provides a bigger area for diffusion to take place across and therefore speeds it up. Let's take a look at both of these diagrams. In these diagrams, food molecules are being absorbed into the bloodstream. In which one do you think this will happen fastest? Well, if we take a look at the one at the top, we can see it's got a very flat membrane surface, and therefore this is a lower surface area than the diagram below, where the membranes are highly folded. So we can call this a higher surface area. Now let's look at the impact that this has. In the top diagram, the molecules will only be absorbed when they're right next to the membrane. In the bottom one, many more of the food molecules are in proximity, and therefore a much higher rate of diffusion will happen. The bottom one is actually a diagram of your small intestines, and they're shaped like this to maximize the food absorption in your body. 6. Osmosis. Osmosis is a special type of diffusion because it involves the movement of water molecules only. And like diffusion, it still occurs down a concentration gradient, with the water moving from a dilute solution, i.e. one that has a high concentration of water molecules, to a more concentrated solution, i.e. one that has a lower concentration of water. An important part of osmosis is that it happens through a partially permeable membrane. This is a membrane that has tiny holes in it, so only very small molecules, like water, can pass through the membrane. But bigger molecules, for example sucrose, can't pass through because they get stuck. Most cell membranes are partially permeable. So, to sum up our definition of osmosis, it is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. It is worth noting that just like diffusion, it is a passive process because it happens down a concentration gradient, and therefore no extra energy is needed. Now let's take a look at osmosis in action. In which direction in this diagram will the net movement of water molecules be? Left to right or right to left? Pause the video and see if you can work it out. Well, to answer this question, we need to identify which side is more dilute and which is more concentrated. The dilute side is the one with the higher concentration of water molecules, which in this case is the left side of the membrane. The right side of the membrane has got less water molecules and more sucrose or solute molecules, and therefore it is the concentrated side of the membrane. So, just to recap, osmosis occurs down a concentration gradient, or from a dilute solution to a more concentrated solution. So this means the net movement of water is from the left side over to the right side. Eventually, the water concentration will be equal on both sides. The water molecules will continue to move back and forth across the partially permeable membrane, but there's no further net movement of water, i.e. the water is balanced on both sides. Osmosis is important in animal cells for making sure that the solutes, like glucose and salts, are at the right concentration inside the cell. This internal environment needs to be kept just right for the cell to work. The difference in concentration between the cell's internal environment and the solution outside it will determine how much osmosis occurs. We're going to look at what happens when a red blood cell is put into 3 solutions that have different concentrations, so you can see the impacts of this on the cell. The first one is a hypotonic solution. This means that it is more dilute than the cell's internal environment. What do you think will happen when the red blood cell is put into this solution? Pause and see if you can work it out. Well, remember that osmosis is the diffusion of water from a dilute solution to a more concentrated solution. So it's going to go from the hypotonic solution into the red blood cell. As the water moves into the cell, it will stretch it, and if a lot of the water moves in, the cell may even burst. This happens if there's a big difference in concentration. Of course, this kills the cell. So next up, we've got an isotonic solution. This means that it is at the same concentration as the red blood cell. Pause again and see if you can think about what will happen when the red blood cell is put into this solution. Well, nothing happens. Because the 2 solutions are the same, there is no net movement of water, i.e. no osmosis occurs. And finally, we've got a hypertonic solution. This means it is a lot more concentrated than the red blood cell's internal environment. Pause once more and see if you can work out what will happen in this case. Well, this time the water will move out of the red blood cell and into the beaker, as the red blood cell solution is more dilute, and the water moves by osmosis to the more concentrated solution. If it's a really big difference in concentration, a lot of water will leave the cell, and this time the cell will shrink, and the same thing happens as when it bursts, it just won't function properly anymore. Now there is a required practical about osmosis that looks at the effect of concentration of salt or sugar on the mass of plant tissue. You'll get to apply your understanding of osmosis a lot more in this practical. 7. Active transport. Using the diagrams on the left, can you work out what active transport is? Pause and have a go. Well, hopefully you've spotted that this time round the arrow is reversed, so substances are moving from where they are in a low concentration to where they're a high concentration, i.e. against the concentration gradient. You might also have spotted that this movement is taking place across a partially permeable membrane, just like osmosis. Because active transport moves substances against the concentration gradient, it requires energy, and this energy comes from respiration. Can you remember the name of this organelle where respiration happens? Yep, it's a mitochondrion. Well done if you remembered that. Before we move on to the definition, we'll look at an example, as it will help us understand the process of active transport in the terms of the keywords dilute and concentrated. So here we've got a plant root, and in the soil around it there are mineral ions. These are in solution, and plants need to absorb them so that they can grow properly. Often plants are in soil that has a low concentration of mineral ions, and because the solute is low, we say it's a dilute solution. Inside the plant roots, however, there are quite a lot of mineral ions, so a lot of solute, or a high concentration, which therefore means it's a concentrated solution. In active transport, the mineral ions move from the dilute solution in the soil into the more concentrated solution in the root hair cells. Moving against the concentration gradient requires energy from respiration in the root hair cells. This allows the plants to continue to grow even when they're in poor quality soil. Returning to our definition, we can now say that active transport moves substances from a more dilute solution to a more concentrated solution, i.e. against a concentration gradient. This requires energy from respiration. Another common example of active transport is in the small intestines. If we zoom in on these, we can see that they are absorbing dissolved food molecules, for example glucose. That's the little yellow molecule there. Glucose is very important as it can be used in respiration to carry out lots of processes in your body. So the body wants to absorb as much of it as possible, and to do this, it uses active transport. If we zoom in a little further, we'll see how this works in the small intestines. So over here, we can see we've got a low concentration of glucose as there is less of it. Therefore, this is the dilute solution. Inside the blood vessels, we've got a high concentration of glucose, and therefore this is called the concentrated solution. Your body is able to move the glucose from where it is in a dilute solution to where it is in a more concentrated solution, and this allows it to move the glucose against a concentration gradient, bringing it inside your bloodstream and transporting it to tissues where respiration can then take place. 8. Mitosis and the cell cycle. To understand the process of mitosis, we first need to understand the nucleus a little bit better. So let's look inside an adult body cell. The nucleus is this little black blob, and if we zoom inside it, we can see it's made up of these structures called chromosomes. There are 23 pairs of chromosomes in an adult body cell, or 46 in total. If we zoom inside a chromosome, we can see that it's actually made up of DNA. This is a molecule of DNA right here. DNA codes for genes. You're going to learn a lot more about this in the inheritance topic in paper 2, but this is all that you need to know for paper 1. Now that you understand that the nucleus contains chromosomes, we can look at mitosis and the cell cycle. The cell cycle is split into 3 parts: Stage 1, Stage 2, and Stage 3. We're going to look at each stage in turn to understand how they work together to produce new cells. Let's start with Stage 1. You can see from the pie chart that Stage 1 is the longest of the 3 stages. In preparation for cell division, it has a lot to do. First of all, the cell will grow, gaining mass. Secondly, it will also increase the number of subcellular structures, such as ribosomes and mitochondria, amongst others. Really importantly is then that the DNA replicates. By doing this, it forms 2 copies of each chromosome. In a human body cell, this would mean that by the end of Stage 1, the cell contains 92 chromosomes instead of 46. We can see this here, that there are 2 copies of each chromosome. Stage 2 is mitosis. In mitosis, one set of chromosomes is pulled to each end of the cell. You can see this happening in the diagram above, with half of the chromosomes having gone to the top of the cell and half of the chromosomes having gone to the bottom of the cell. As well as this, the nucleus will also need to divide in 2, splitting the chromosomes in half. You can see this here, that the nucleus is starting to pinch in, and that will completely split off. And finally, in Stage 3, the cytoplasm and cell membranes divide to form 2 identical cells. These cells are genetically identical to each other and also to the parent cell, which was the initial body cell. Just a quick note to mention that if you take A-level biology, you'll learn about mitosis in a lot more detail. If you've come across this already, then remember that you don't need this level of detail at GCSE, you just need what I've already covered. You're probably wondering what the importance of mitosis and the cell cycle actually is. Why do we need new cells? Well, there are 3 main reasons. The first is development. When you're first conceived, you're just a single cell. You're going to need to make a lot of new cells for you to develop all the way from being an embryo to an actual little baby. Secondly, during childhood and puberty, you need the ability to grow, and therefore you need to be able to make new cells. And finally, sometimes we do need to repair our cells. Imagine you got a graze and you're going to lose some of your skin. Your body can therefore make new skin cells through mitosis to repair the damage. As you can see, It's a really important process. 9. Stem cells. We're going to start with recapping what differentiation is. We looked at this in the specialized cells video. When a cell differentiates, it means it changes by acquiring different subcellular structures like mitochondria and ribosomes. By doing this, it becomes a specialized cell. Can you name these 3 specialized cells below? On the left, we've got a nerve cell. In the middle are muscle cells, and on the right is a red blood cell. This unspecialized cell is a stem cell. A stem cell is an undifferentiated cell, and they've got 2 main functions. The first we've already covered, which is differentiation to produce different cell types. And the second function is that it is capable of giving rise to many other stem cells, i.e., it can regenerate new stem cells. We're going to look at the uses of stem cells in animals and plants. Let's start with animals. Most animal stem cells will differentiate at a very early stage. This means that more stem cells can be found in an animal when it is still an embryo. We call these embryonic stem cells. An embryo forms when a fertilized egg cell divides, and this forms a hollow ball of cells. On the inside of this, we find the embryonic stem cells. The function of these is to differentiate to make all of the specialized cells in the body. But scientists have come up with another use. Embryonic stem cells can be cloned. They can then be induced to differentiate into lots of different cell types, like the ones that you see here. And these can then be used for the treatment of conditions where cells in the body aren't working properly. For example, paralysis is when nerves are damaged. The body can't make new nerve cells on its own, so using stem cells to make more could potentially cure paralysis. Similarly, type 1 diabetes is when cells of the pancreas are damaged. Replacing them with working pancreatic cells could cure this condition. There are other applications as well, like new heart cells to repair heart damage. But paralysis and diabetes are the 2 conditions that you need to learn for this specification. Although most animal stem cells differentiate at the early stage of development, there are some that remain into adulthood. We call these adult stem cells. One place where adult stem cells are found is in the bone marrow. These stem cells can differentiate to form many other types of cells, including blood cells. For example, they could differentiate to make white blood cells and red blood cells. Adult stem cells are more limited in their uses, as they tend to only make a small number of different cell types, whereas embryonic stem cells can make almost any cell in the body. Now on to plant stem cells. Plant cells can differentiate throughout their entire life, as they continue to grow for their whole life. Meristem tissue is found in the shoots and root tips of plants, and this is where you'll find the stem cells. You can see down here that a lot of mitosis is taking place, and this actually happens constantly so that the plant is continuing to grow. Because plant stem cells can differentiate into all cell types even in adulthood, we can use them to clone plants. This has got some real advantages. The first is that using stem cells to clone plants is very quick and economic, i.e. cheaper than other methods. For example, the bananas that we eat are all cloned from a single plant. Cloning plants for commercial sale is also known as horticulture. We can also use stem cells in cloning rare plants, saving them from extinction. As you'll see in the ecology topic, extinction is an ever-growing concern in our world, so the ability to save rare plants from this is very important. A big benefit for farmers is the ability to clone crops with special features like disease resistance. And lastly, cloning large numbers of genetically identical plants for research is really useful. As the plants are all the same, the effects of the research procedure can be tested more easily. Finally, we need to look at the issues surrounding stem cells. Adult stem cells have a risk of carrying viruses, which could infect the patient receiving them. And if a patient receives stem cells from an unrelated person, there's a chance that their body will reject them. They'll have to use immunosuppressant drugs to prevent this. One solution to this is therapeutic cloning. In this process, cells from the patient are used to create an embryo, which then has the same DNA as the patient, so there's absolutely no rejection risk. The embryonic stem cells could then in theory be used to treat conditions and even grow entire organs. This process is still in the research stages, but there are high hopes for the future of stem cells in medicine. Other issues of stem cells include ethical issues. Embryos cannot give consent to be used in research, so some people think it is against their human rights to use them. One counter-argument is that most of the embryos used are left over from fertility treatments and would otherwise be discarded. Another issue is that it is against some people's religious beliefs, as they think we shouldn't interfere with the natural reproduction process at all. In some countries, the use of embryos in research is banned. Here in the UK, it's allowed, but there are some very strict regulations around it. And finally 10, culturing microorganisms. Bacteria multiply by a type of simple cell division, which is called binary fission. It's very similar to normal cell division. First up, the genetic material replicates. In this case, it's a loop of DNA as we're talking about bacteria. The two copies then move apart to opposite ends of the cell. Next, new cell walls form. And in addition to this, the cytoplasm will divide. Eventually, when the walls form in the center, they split off into two cells. You'll notice that there are plasmids, and these aren't split equally between the two cells, so each cell is slightly different. You can remember the name binary fission by thinking of it like this. Bi means two, like a bicycle has two wheels, and fission means splitting. You may have learned about this in physics when looking at nuclear fission. So, putting the two together, binary fission means splitting in two. It's a very quick process, and it can take as little as 20 minutes for the bacterial cell to split in two. That's provided it has enough nutrients and a suitable warm temperature. You need to know how to calculate the number of bacteria in a population. You'll be given the mean time and asked to work out how many there are after a certain length of time. So let's look at an example. Staphylococcus aureus divides once every 30 minutes. What is the population size after 3.5 hours? The first step is to check that our units are the same. At the moment, we've got minutes and hours, and they need to be the same. So let's turn 30 minutes into hours, which is 0.5 hours. You get this by doing 30 divided by 60, or just think of it as half an hour. Next, we'll work out how many times it has divided. So to get the divisions, we're going to do the time that we've been given. In this case, it's 3.5 hours, and we divide it by the length of one division, which in this case is 0.5. So we take our numbers and we pop them in, and 3.5 divided by 0.5 gives us 7 divisions. So that's 7 divisions in 3.5 hours. It's important to remember that each new cell that is created will also divide in this time. So each new cell will divide to give 2 new cells. Because of this, we're able to use the number 2 to work out our answer, and we just do 2 to the power of the number of divisions. So in this case, it's going to be 2 to the power of 7. This just means you're multiplying 2 by itself 7 times, but it's easier to do it on your calculator, and you get 128. Okay, try the next question on your own, and then press play when you're ready to go through the answer. Ready? So, Helicobacter pylori divides once every 45 minutes. Calculate the population of the colony after 9 hours. So again, our units aren't the same, so we're going to turn the minutes into hours. And that gives us 0.75 hours when we divide 45 by 60. Then we're going to work out the number of divisions. We're going to divide the time given, which here is 9 hours, by the length of the division, which we just worked out, was 0.75 hours. And when we pop that into our calculator, it gives us 12 divisions. So we take our magic 2 and put it to the power of 12. From our calculator, we get 4,096. Now you could be asked to give your answer in standard form. This is a higher tier skill and it's one of the mathematical parts of your paper. So pop on over to my math skills and biology if you haven't learned how to do this skill yet. When you're done, come back here and see if you're now able to do it. Okay, so what we're going to do is make a number that's between 1 and 10 by placing a decimal point somewhere in this number. That would go here to give us 4.096. And now we're just going to work out what our 10 is to the power of. So we count 1, 2, 3 back to the original decimal place, and that's going to give us 4.096 times 10 to the 3. And remember, that's higher tier only. Now we're going to look at how you can prepare uncontaminated bacterial cultures using aseptic technique. For the aseptic techniques, you need to be able to explain why those steps are done, so I'll do those parts in blue. Firstly, bacteria are grown in a culture medium that supplies them with nutrients to grow. This can be supplied by either a nutrient broth or a solid agar jelly. These are both found inside a petri dish. The petri dishes and culture media must be sterilised before use. This ensures that any unwanted microorganisms are killed. This wire loop is called an inoculating loop. Just like before, the inoculating loop must be sterilised before use by passing it through a hot flame, like a Bunsen burner flame. This, of course, kills any microorganisms that were on it. It's then allowed to cool down and then used to spread your bacteria. After transferring and spreading the bacteria, the lid of the petri dish should be secured lightly with adhesive tape. This is important as it prevents any microorganisms that are in the air from entering your petri dish and culture. The petri dish is then stored upside down. This looks odd, but it stops any drops of condensation forming on the lid and dropping down onto the agar surface, which could damage or prevent growth of the bacterial cultures. Finally, in school laboratories, cultures should be incubated at 25 degrees Celsius or lower. If a higher temperature were used, harmful pathogens would be more likely to grow, so 25 degrees Celsius and lower is safer for schools. In industrial conditions, cultures may be incubated at higher temperatures so that they grow faster. Now, there is a required practical on the culturing of microorganisms, and in this you'll look at the effect of antiseptics or antibiotics on bacterial growth. You'll learn more about zones of inhibition and calculations for this then, so we won't cover it here. Our second topic is organisation, and we're starting with number 1, principles of organisation. Like many things in life, there's a natural hierarchy to the organisation of living things. In the last topic, you learned all about cells. Cells are the basic building blocks of life, and they are first in the hierarchy. Examples include sperm cells, nerve cells, which are also called neurons, and root hair cells, amongst others. Next up are tissues. A tissue is a group of cells with a similar structure and function. Let's look at an example. Here we've got muscular tissue, and its function is to contract, causing movement. And you can see that all of the cells have a similar structure. Then we have organs. These are collections of tissues performing specific functions. For example, the stomach here has got 3 different types of tissue. Firstly, we've got muscular tissue to churn and move the food. We've got glandular tissue over here that can release digestive juices. And the stomach has got epithelial tissue. This covers the surfaces of the stomach, both inside and out. Lots of organs combine to form an organ system. The organs in an organ system work together to carry out a specific function. For example, down here we've got a diagram of the digestive system. The digestive system organs are all working together to digest food. You've also got a respiratory system, and these organs work to bring oxygen into the body and remove the CO2 from it. Let's take a look at the final hierarchy. At the bottom, we've got cells, which build up into tissues, which build up into organs, and lots of organs make an organ system. And what do you think is at the top? Yep, it's the whole organism itself. For example, a human. 2. The human digestive system. The process of digestion is really important because the food that we eat can't be absorbed immediately. It's too large and insoluble. But digestion breaks it down into smaller soluble molecules, which can then be absorbed into the bloodstream. The GCSE specification assumes knowledge of the digestive system from Key Stage 3 science, so it's important that we recap that first. Let's start by going over what each organ does. To start, we've got the mouth. This takes in the food and mechanically breaks it down with the teeth. It then gets mixed with saliva. Speaking of saliva, over here we've got the salivary glands. These are what produce the saliva. The food then gets swallowed and passed down through the esophagus. This tube transports food to the stomach. The stomach over here is where the food is then churned and mixed with hydrochloric acid. It's important that you learn the name of this acid, so we'll highlight it here. It then passes into the small intestine. Here, it continues to digest the food and then absorbs the resulting small soluble molecules. We're going to take a minute to see how the small intestine is adapted for its function, as this is one of the exchange surfaces you need to know. There are three main adaptations to learn. Firstly, the small intestines have a very large surface area. This is achieved by having villi. If you zoom in on the small intestines, you'll see the villi, and each one is called a villus, that's the singular. These are just folds in the surface of the small intestine. The large surface area is also achieved by microvilli. If we zoom in on just one villus, we can see it's covered in loads of microvilli. These folds on the surface of the villi really increase the surface area. Secondly, the small intestines have a good blood supply. There are loads of capillaries supplying the villi. This ensures that any food molecules absorbed are quickly taken away to other tissues, maintaining a steep concentration gradient, which results in faster diffusion. This is also covered in the diffusion video. Thirdly, the small intestines have got a short diffusion distance. This is the distance from the food molecules to the bloodstream. As the food molecules don't have to travel very far, this means it's a faster rate of diffusion. Okay, let's carry on with our journey through the digestive system. After the small intestines, the food passes to the large intestines. This is where water is absorbed from any undigested material. We call this undigested material feces, and it gets stored in the rectum. Then finally, it gets excreted through the anus. Now we're going to add the new bits that you need to know for GCSE level. So we mentioned the salivary glands produce saliva, but they also produce a digestive enzyme called amylase. You'll learn a lot more about enzymes in the next 3 videos. The stomach also produces a digestive enzyme, and this one is called protease. Now we've got a new organ called the pancreas. This small organ produces all 3 digestive enzymes: amylase, protease, and lipase. And over here, we've got the liver. It's a large organ that produces bile. Bile helps by speeding up enzyme action. We'll talk a lot more about bile in the video Factors Affecting Enzymes, so look out for that. Finally, we've got the gallbladder, and this is where the bile is stored before it's finally released into the small intestines. 3. Enzymes, lock and key theory. Catalysts speed up the rate of chemical reactions, and they do this without being used up or changed themselves. Because of this, they can be used again and again. Enzymes have a similar function. They are biological catalysts, and they speed up the reactions in living organisms. They can be used again and again as well. They're actually just a special type of protein. All proteins are made from chains of amino acids. Amino acids are small organic molecules, and you'll learn more about them if you take A-level chemistry. The folding of the amino acid chains is what gives a protein its specific shape and structure. In enzymes, this folding is particularly important. It determines the shape of the enzyme's active site. Let's look at what this actually is. Enzymes are like a padlock, and the active site is the keyhole. It's got a specific shape, and this is really important as it's going to allow just one key to fit inside it. The key is called the substrate. This is a word you need to learn. Because enzymes and substrates work together like a lock and a key, we call this the lock and key theory of enzymes. Okay, so now let's take a look at an example. One example of a substrate is starch, and the enzyme that it binds to is called amylase. So if they come into contact with each other, the starch will bind to the active site of the amylase. When this happens, the amylase catalyzes the reaction, which means it speeds it up. And in this case, the reaction taking place is the breakdown of the starch into its products. And starch breaks down into glucose. So here you can see we've got 2 glucose molecules, and those are the products. Different enzymes have different active site shapes. This is because their amino acid chains have folded differently, giving a different 3D structure. Because enzymes affect the rate of reactions, they affect the metabolism of a cell or organism. This is a term that comes up in the specification under the fundamental concepts and principles of biology section. We'll cover it again in the bioenergetics topic, but for now, here's the definition for metabolism. It is the sum of all of the reactions that take place in a cell or organism. Hopefully this little picture will help you remember it. By affecting the speed of reactions, enzymes control the metabolism of a cell or organism. Now, it's important to note that not all enzymes break down substrates. Some enzymes are able to catalyze reactions that build large molecules from smaller molecules. Other enzymes can change one molecule into another. 4. Factors affecting enzymes. I've mentioned before that enzymes are a special type of protein that can catalyze reactions in living organisms. They do this by having an active site that is a specific shape for a substrate molecule to bind to. Keeping this specific shape is vital to the enzyme working properly. If the active site changes shape, the substrate can no longer bind to it. There are 2 factors that can alter the shape of the active site, and therefore affect enzyme function. The first is temperature. This graph shows how temperature affects the rate of reaction, and you need to be able to explain every part of it. Just like in normal reactions, increasing the temperature will increase the rate of reaction. This is because the enzyme and substrate molecules have got more kinetic energy, resulting in more successful collisions, and therefore more reactions. The optimum temperature is where enzyme activity is at its highest. In humans, this temperature is around 37 degrees Celsius, as this is the temperature at which our enzymes work best. In other organisms, it can be different. Increasing the temperature beyond the optimum temperature will cause the enzyme activity to decrease. This is because the enzyme has been denatured. Let's take a moment to see what this word means. Hopefully you recall from the lock and key theory video that enzymes are made from chains of amino acids that have been folded to give them their specific shape. This structure is actually held together by weak forces between the amino acids. High temperatures can break these forces, causing the amino acid chains to change shape. This alters the shape of the active site, which we call denatured. This is an important word to learn. When the enzyme is denatured, the substrate molecules can no longer bind to the active site, and the reaction will slow down or stop altogether. Enzymes are also affected by pH. This is how acidic or alkaline their environment is. Each enzyme will have an optimum pH, and this can vary in the digestive system. For example, the protease produced in the stomach has a low optimal pH. The stomach secretes hydrochloric acid to maintain the acidic environment so the protease can function efficiently. Changing the pH too far above or below an enzyme's optimal pH can also denature the enzyme. It does this by changing the shape of its active site. Changing the pH affects the charges on amino acids, and this changes their folding structure. Maintaining the right pH is important for efficient digestion. Speaking of efficient digestion, let's talk about bile. In the digestive system video, We mentioned that bile is produced in the liver, stored in the gallbladder, and acts in the small intestine. Now let's look at what it actually does. Enzymes produced in the pancreas, over here, and the small intestine, over here, work best in alkaline conditions. When food leaves the stomach and enters the small intestine, some of the hydrochloric acid comes along with it. Bile is an alkaline liquid, so it is able to neutralize this HCl that comes from the stomach. This maintains the right alkaline conditions needed for the enzymes in the small intestines to work properly. Bile has another function. It helps with the digestion of fats by emulsifying them. This means that they're physically broken down from larger droplets into smaller droplets. This provides a much larger surface area for enzymes to act upon, and this increases the rate of digestion. Hopefully you can see this in the diagram here. It's important to note that bile is not an enzyme, as it isn't chemically breaking down the fat molecule into different molecules, it's just physically making it into smaller pieces. 5. Digestive enzymes. In the Digestive System video, you heard the names of 3 digestive enzymes. Today, we're going to look at what they break down and the products of this breakdown. So let's take a look at the names of them. First up, we've got amylase, and this is an example of a carbohydrase enzyme. What do you think carbohydrases break down? Taking a look at the picture, it's things like bread and pasta, which we call carbohydrates. Amylase in particular breaks down the carbohydrate that's called starch. You may have heard of this already. And the products of this are simple sugars, for example, glucose. Our next enzyme is protease. What do you think it breaks down? Well, the start of the word is a hint. They break down proteins, for example, these chickpeas. Now, for the products, see if you can remember what proteins are made from. Well done, if you remembered, they're made from amino acids, so when they're broken down, they turn back into amino acids. Our third enzyme is lipase. Any ideas of what it breaks down? Well, the start of the word is another hint. It's lipids, otherwise known as fats and oils. For example, this cheese. The products of lipids are fatty acids and glycerol. As well as knowing what each enzyme does, you also need to know where they are produced and the site of action, i.e. where they work. I'm going to start with a top tip for you. All 3 enzymes are produced in the pancreas. Let's change those into green to match the colour of the pancreas. Another top tip is the site of action for all 3 enzymes is the small intestines. That means they all work in this organ. This is really good to know for any exam questions. Okay, but they do also get produced in other places. Amylase, for example, also gets produced in the salivary glands. It's then no surprise that the site of action is the mouth, as that's where the salivary glands are located. Protease, on the other hand, is produced in the stomach, and therefore it also works in the stomach. And it's also produced in the small intestines. Lipase is also produced in the small intestines. Now, different to the others, its only site of action is the small intestines. And this kind of makes sense when you look at the fact that the pancreas empties its digestive enzymes into the small intestine, and that's the only other place that they are made. So that's the only place where it works. 6. The blood. The blood is part of the human circulatory system, along with the blood vessels and the heart. You'll learn more about these in separate videos. There are 4 components of the blood. You need to know what each part does and what it looks like. It may surprise you that over half of your blood isn't actually made of cells. It's made of plasma. Plasma is a pale, straw yellow coloured liquid. Its function is to transport your blood cells around the body, along with some other chemicals such as carbon dioxide, urea, and proteins. Then we've got red blood cells. These have the important function of carrying oxygen around the body, bringing it to the cells that need it. To do this, they have some special adaptations. First up, they've got a pigment called hemoglobin. This can bind strongly to oxygen, which helps them hold on to it and carry it around the body, It's also what makes the red blood cells red. Secondly, they have no nucleus. This allows more space for the hemoglobin pigment. And thirdly, they're biconcave, which means they have a dip on each side. This increases their surface area to volume ratio so they can carry more oxygen. Red blood cells make up around 45% of the blood, and the remaining less than 1% are white blood cells and platelets. White blood cells are covered in the infection and response topic. You'll go over them in a lot more detail then. So for now, we'll just cover their main function, which is to protect the body against infection. Unlike red blood cells, they do have a nucleus, and they're normally larger. They're also not as strongly pigmented, so if you're asked to identify them in a photograph, they'll just be the colour of whatever stain has been used. Finally, we've got platelets. These are fragments of cells, and their function is to help the blood clot. This is important when a person gets wounded, as it helps prevent blood loss and forms a scab that prevents bacteria from entering the body. As they're fragments of cells, they're quite a lot smaller than red or white blood cells. So this is how you can identify them. 7. Blood vessels. There are 3 types of blood vessels. You need to know the structure and function of each. The arteries carry blood away from the heart, and I remember this with A for away. They're carrying it to the organs of the body. The blood is under high pressure as it's just being pumped out of the heart. Because of this, the arteries have thick muscular walls with elastic fibers, and this allows the walls to stretch as the blood is forced through them. This is actually what you're feeling when you take your own pulse. Over here, the hole in the middle is called the lumen. Arteries have a relatively small lumen, and that keeps the blood under high pressure. The arteries will eventually branch out into capillaries. Capillaries are tiny blood vessels that you can't actually see with the naked eye. Their job is to bring the blood to the cells of the body. Once they've done that, substances can then easily diffuse out of the blood and into the cells. For example, oxygen and glucose may diffuse into the body cells, and waste products like carbon dioxide will diffuse out of the body cells. Capillaries have very thin walls, and you can see that they're just 1 cell thick. This short distance makes diffusion much easier and therefore faster. The blood will then pass from the capillaries to the veins. They carry the blood from the body's organs back to the heart. At this point, the blood is under lower pressure. This means that veins can have thinner walls as they don't need to maintain a high pressure. They also have a wider lumen to allow the blood to flow easily through it with less resistance. And they've got valves. These allow the blood to flow forward to the heart but prevent any backflow of blood, which is really important. 8. The lungs and gas exchange. In the previous video on blood vessels, we saw that capillaries allow oxygen to diffuse out of the blood and into the body's cells where it's used, and they allow carbon dioxide to diffuse out of the cells and into the blood. This CO2 needs to be removed from the blood and the oxygen needs to be replenished. This happens in the process of gas exchange, which takes place here in the lungs. You need to know the structure and adaptations of the lungs, so let's start by labeling this diagram. Why not pause and see how many parts you can already name? Well, up here we have the nose and the mouth, and those bring air into the body. And then here we have the trachea or the windpipe. This has rings of cartilage to keep it open. It then branches into the bronchi, or bronchus for singular, which then itself branches into bronchioles. At the end of the bronchioles, we've got tiny little air sacs called the alveoli. We'll look at these more in a second. Over here, we've got the ribs. They make up the rib cage, which protects the lungs. And between them are intercostal muscles. These help move the rib cage. And finally, down here, we've got the diaphragm. This muscular tissue also helps with breathing. Now let's look at the adaptations for gas exchange. Here is one alveolus. This is where gas exchange takes place. And you can see that it is surrounded by a capillary. The first adaptation is that it is ventilated. This means the air is actively brought in and out of the lungs.
This helps maintain a steep concentration gradient as fresh, oxygen-rich air is brought into the lungs with each breath. Gas exchange then takes place, and the CO2 that entered the alveolus from the blood will then leave with each exhaled breath. Secondly, it has a good blood supply, as it's surrounded by a capillary network that covers 70% of the alveolus. Let's look at what's happening in the blood. The blood that arrives to the alveolus has got a high concentration of CO2. This diffuses out of the blood and into the alveolus, where it has a lower concentration of CO2. The blood that is arriving also has a low concentration of oxygen, whereas the alveolus has a high concentration of oxygen. That means it diffuses out and into the blood. This maintains a really steep concentration gradient, as the blood quickly transports that new oxygen away, and that keeps that diffusion gradient going. Our third adaptation is that the walls of the alveoli are very thin, providing a short diffusion pathway between the air and the blood, so it's much faster. If we take a look at the shape of the alveolus, we can see it's a spherical shape and it's tiny, so they have a large surface area to volume ratio. This maximizes the rate of diffusion. Now that we've looked at gas exchange in humans, we're going to briefly look at it in fish. You may have already covered this with your teacher in Topic 1 when learning about diffusion. Fish get their oxygen from water, which only holds very low concentrations of oxygen. They therefore need a really efficient gas exchange process. Here's how it works. Water enters through the mouth of the fish and then passes over the gills, which are inside the body. It then leaves by a special flap called the operculum. Gas exchange happens in the gills. The diagram below shows you what the gills are made of. These are stacks of filaments, and these help provide adaptations. The first is that the filaments provide a large surface area. They also have a good blood supply with lots of capillaries along them. The gill filaments and capillaries are just one cell thick. That provides a shorter path for diffusion. And they are ventilated, as water containing oxygen is actively moved over the gills. And finally, they maintain a counter current. This just means that the blood moves in the opposite direction to the flow of the water. This is good because it maintains a steep concentration gradient for diffusion. Hopefully you can spot that there are some similarities between gas exchange in the human lungs and the gills of the fish. 9. The heart. The heart is an organ that pumps blood around the body in a double circulatory system. This means that each side of the heart pumps the blood to a different place. The right side of the heart has deoxygenated blood, so it needs to pump the blood to the lungs. And up here you can see the capillary network of the lungs. This is where gas exchange takes place, providing the blood with oxygen. The oxygenated blood then returns to the left side of the heart, which pumps this blood to the rest of the body. And down here you can see the capillary network of the rest of the body. In the capillaries, oxygen can diffuse out of the blood and into the body's cells, where it's used for respiration. Carbon dioxide is produced, and this will get transported up to the right side of the heart to be taken to the lungs. The double circulatory system works really well, as it means the oxygenated blood is pumped separately, and so it's sent under a really high pressure so it can reach all parts of the body. Now let's look at the structure of the heart. You'll notice that it says right side over here on the left side of the diagram, and it says left side on the right side of the diagram. When we look at a diagram of the heart, the sides are flipped. This is because it's labelled as though it's facing you. The heart has 4 chambers. The top 2 chambers are called atria, so we've got the right atrium and the left atrium. And the bottom chambers are called the ventricles, so we've got the right ventricle and the left ventricle. My tip to remember which way round the chambers are is that the bottom chambers look a little bit like a V for ventricle, and then the top chambers you just need to remember are the atria. The blood comes from the body and it enters the heart through a vein called the vena cava.
You can remember this by V for vein is V for vena cava. Remember, veins always bring blood back to the heart. The blood then travels down through the right atrium into the right ventricle, where it gets pumped out through an artery called the pulmonary artery. Remember, arteries take blood away, and in this case, they're taking it to the lungs. Gas exchange takes place there, and the oxygenated blood returns to the heart by the pulmonary vein. Pulmonary just means that it's relating to the lungs. The blood then travels down through the left atrium and the left ventricle before getting pumped out of the heart and through the aorta. The aorta is a major artery and it takes the blood to the rest of the body. One way to remember this blood vessel is that it's A for artery and A for aorta. Now down here you'll notice that the left side of the heart has got thicker muscle tissue than the right side. This allows it to pump the blood under a higher pressure to make sure that it gets to all the parts of the body that it needs to. The last thing to note is that we've got some special cells here in the right atrium. These cells are called the natural pacemaker cells of the heart. They release an electrical signal that causes the heart to contract, and this then pumps the blood out of it. These control the resting heart rate of the heart, also known as your pulse. 10. Coronary heart disease. The heart pumps blood to all of our body's cells. It works very hard and requires lots of oxygen and glucose for its own cells. These are supplied by the coronary arteries, and you can see them here covering the heart. Sometimes the coronary arteries can become narrow, usually caused by a buildup of fatty deposits. This is called coronary heart disease, or CHD. This is dangerous because the fatty deposits restrict the blood flow to the heart. Less blood flow means less oxygen is being supplied, which can cause pain and even heart attacks. There are 2 methods of treatment for CHD. You need to be able to evaluate methods of treatment, so we'll discuss the benefits and risks as we go through them. The first method is stents. These are little mesh tubes that can be put inside the blocked artery, where they then squash down the fatty deposit, opening the artery wider. They are left in place, allowing the blood vessel to work properly. The issues are that the stents can cause a risk of clotting after they've been put in. The second method is statins. These are a drug that lower blood cholesterol levels, slowing the buildup of fatty deposits. They don't unblock any blocked arteries, so they're generally only used for people who are at risk of CHD. Besides the main benefit of reducing the buildup, they also lower the risk of some other diseases. But as they are tablets, people need to remember to actually take them, and they're a little bit slower to have an effect. There are also some possible side effects that are quite serious. Now we'll take a look at some other cardiovascular diseases. In some people, the heart valves may become faulty, particularly in older people. This means they may not open fully, or they could be leaky. When this happens, the blood can flow in both directions, so that less oxygenated blood is pumped to the tissues of the body. This results in people feeling breathless or tired, as their cells aren't receiving enough oxygen for respiration. Without treatment, faulty valves can result in death. Fortunately, doctors can replace the faulty valves with either mechanical valves or biological valves. Mechanical valves are man-made, and the good news is that they last forever. The bad news is that patients will need to take medication to prevent any blood clotting forming around the valves. Biological valves, on the other hand, are valves from donors, so the patients don't need any medication, but they only last for around 10 to 15 years. Next up are pacemakers. In the last video about the heart, you will have learned that the natural pacemaker cells of the heart are in the right atrium, but sometimes these cells aren't functioning properly. They can make the heart beat too fast or too slow. In both cases, this causes problems. So if the heart is beating irregularly, an artificial pacemaker needs to be implanted into the chest. This is attached to the heart with 2 wires, and then it can release an electrical signal which causes the heart to contract at the rate that's needed. These devices are tiny and life-saving. If a patient has heart failure, they may require a heart transplant or a heart and lungs transplant. This requires a donor heart, which isn't always available. Doctors may use an artificial heart to try and keep the patient alive until a donor heart is found. The big advantage of this is that it's less likely to be rejected by the patient's body, and of course, it prolongs their life. But it can cause the blood to stick and clot, leading to strokes. And to prevent this, the patient needs to take blood thinners, which have their own complications, like extended bleeding if they're in an accident. There's also a lot of machinery involved in keeping the artificial heart working. That means that patients need to stay in hospital until a donor heart is found. Natural heart transplants work a lot better, and there's a much lower chance of the blood clotting. It also allows the patients to live a more normal life. But as with any organ transplant, there's a risk that the heart will be rejected, as the patient's body may detect it as foreign. 11. Health issues. Health is a state of physical and mental well-being. It's important to remember that it is both physical and mental, not just one or the other. Diseases are major causes of ill health, and they can be split into two groups: communicable and non-communicable. Communicable means the disease is infectious and can be passed on from one person to another. Non-communicable means it cannot be transmitted. You'll learn about some non-communicable diseases in the next video, and you'll find out about communicable diseases in the infection and response topic. Other factors affecting physical and mental health include diet, stress, and difficult life situations. Diet could mean a lack of food or certain nutrients. This can cause diseases like anemia, scurvy, and starvation. Or it could mean too much food or too much of specific unhealthy foods. This can cause conditions like type 2 diabetes and obesity. We all experience stress, and some of this is okay, but high levels of it can affect mental health and even increase a risk of certain diseases like heart disease. Life situation includes things like income, where in the world you live, access to healthcare and clean water, etc. Many people have little or no control over their life situation, and it can have a big effect on mental and physical health. Different types of diseases may interact with each other. There are 4 examples that you need to know. Defects in the immune system mean that an individual is more likely to suffer from infectious diseases. These defects could be caused by genetics or by something like HIV. Viruses living inside body cells can be the trigger for certain cancers. For example, the human papillomavirus, or HPV, can trigger cervical cancer. Immune reactions initially caused by a pathogen can trigger allergies, such as skin rashes and even asthma. Severe physical ill health can lead to depression and other mental illnesses, particularly if they affect the person's ability to do everyday activities. For example, if somebody is bedridden. Now, you need to be able to use graphs, frequency tables, bar charts, histograms, and scatter diagrams to analyze information about diseases. These skills are all covered in my Maths and Biology video, and they can come up in other topics. I suggest you take some time to practice these skills, as it's quite likely they'll come up with this topic. 12. Non-communicable diseases. Non-communicable diseases are ones that aren't infectious. This means they can't be transmitted from person to person. These diseases cause 71% of deaths globally, which far outweighs the number of deaths caused by communicable diseases, at just 29%. Both of these diseases, not just communicable diseases, affect the individual, their family, which is supporting them, and their country. Countries spend huge amounts of money trying to treat ill people. And not only that, it affects the global economy because people who are ill aren't able to contribute to the workforce. What's crazy about these diseases is that most of them are preventable, as they are caused by some lifestyle factors like smoking or a lack of exercise, or they can be caused by substances in the person's body or environment. For example, UV light that comes from the sun. Lifestyle choices and substances that increase the risk of disease are called risk factors. They increase the rate of disease incidence, which just means how many people have the disease. Now, sometimes there is a correlation between something like a lifestyle factor and a disease. For example, here the number of ice creams eaten per 100 people increases, as does the incidence of sunburn per 100 people. But just because there's a correlation doesn't mean there's a causation. We know that eating ice cream doesn't make you sunburnt. It just so happens that eating ice cream and getting sunburnt both happen on sunny days. To find out if certain factors really cause diseases, doctors and scientists need to do a lot of research to understand the biological mechanism causing this. When this is proven, we call it a causal mechanism. A causal mechanism has been proven for some risk factors, but not in others. There are 6 proven causal mechanisms that you need to know. The first is cardiovascular disease, which has the risk factors of diet, smoking, and exercise. A poor diet can increase the levels of blood cholesterol. Smoking can increase the blood pressure and damage the lining of the arteries. And a lack of exercise can lead to a buildup of fatty deposits in the arteries. Type 2 diabetes can be caused by obesity. The result of this is the body stops responding to an increase in blood glucose levels, which is dangerous. Liver and brain damage are both caused by alcohol. This can cause liver cancer and cirrhosis, which is scarring of the liver. It can also cause brain damage, which stops the brain functioning properly even when sober. Lung disease and cancer are both caused by smoking. Tar is a carcinogen, and it turns the lungs grey and increases the risk of lung cancer. And damage to the fetus is caused by both smoking and alcohol. The results of this can be a low birth weight, a premature birth, or even a stillbirth. This is when the baby is born dead. Now, cancer is something you're going to learn about in the next video, but for now you just need to know that it's an uncontrolled cell division. It can be caused by carcinogens in a person's environment, including ionizing radiation like UV light from the sun, and radioactive metals like uranium. It can be other substances as well, like tar, or possibly even an excess of red meat in the diet, which is linked to bowel cancer. Many non-communicable diseases are caused by multiple factors interacting, so it can be hard to find a causal mechanism. 13. Cancer. Cancer occurs when changes in a cell cause abnormal or uncontrolled cell growth and division. This can lead to the formation of tumours in the body, like the one you see here. Benign tumours are contained in one area of the body, usually within a membrane. They don't spread to other parts of the body. However, they can still be dangerous as they grow very quickly and can put pressure on other organs, which damages them. This happens if there's no space for them to grow into. For example, tumours in the brain have nowhere else to grow as the skull surrounds them, so they can put pressure on the brain, which is very dangerous. So these types of tumours can still be life-threatening. Malignant tumours are different. Small parts of them can break off, and these cells can travel in the blood. This allows them to spread to different parts of the body, and they can also invade neighbouring tissues. Once there, they can then cause secondary tumours. Malignant tumours are difficult to treat because of how they spread around the body. The 2 main methods of treating cancer are radiotherapy and chemotherapy. You don't need to know the details of these, so we'll just look at this very briefly. Radiotherapy uses radiation to target and kill cancer cells, but it can harm healthy cells too. Chemotherapy uses chemicals. These can be taken either through tablets or through a vein in the hand or arm. These chemicals work by halting mitosis in rapidly dividing cells, i.e. cancer cells. Unfortunately, cells like your hair cells are also rapidly dividing, which is why patients undergoing chemotherapy can often lose their hair. Both treatments have many side effects, and the results can be quite variable from person to person. There are certain lifestyle factors that increase the risk of different cancers. For example, we have smoking, obesity, exposure to UV light, and common viruses like HPV. All of these will increase the risk of a specific cancer. For example, HPV increases the risk of cervical cancer. There are also genetic risk factors for cancers, for example, breast cancer and ovarian cancer. 14. Plant tissues and organs. Plant organs include the root, stem, flowers, and leaves. We're going to look at the tissues of the leaves and how they help it carry out photosynthesis. This is a cross-section diagram of a leaf, so you can imagine that we've cut through it and are looking at it from the side. So first up, we have the epidermal tissues. We have got the upper epidermis and the lower epidermis. These both cover the surface of the plant and protect it. They can also secrete a waxy substance, which we call the waxy cuticle. This waterproofs the leaf, which minimizes water loss. You'll also notice that the upper epidermis is quite thin. This allows light to penetrate through to the next layer. The next layer is the palisade mesophyll. This tissue is responsible for photosynthesis. You can remember it by P for palisade as P for photosynthesis. To carry out this function, it has lots of chloroplasts, and these contain chlorophyll to absorb the light for photosynthesis. This tissue is tightly packed, and this maximizes the amount of light that gets absorbed for photosynthesis. The next tissue is the spongy mesophyll. Just like a normal sponge, it has lots of air spaces. This allows gas exchange via diffusion to take place easily. This is important as carbon dioxide needs to diffuse into the plant cells for photosynthesis, and oxygen that is produced needs to be able to diffuse out. On the underside of the leaf, or the bottom of the leaf, we've got tiny little pores called stomata. Stoma is a singular for just one of these little holes. The size of this hole is determined by guard cells that are found on either side of the stoma. These are important for controlling water loss, which you'll learn more about in the next video. Now we're going to look at the transport systems of the plant. This consists of the xylem and the phloem. The xylem transports water and mineral ions. These are absorbed from the soil in the roots and are then transported up through the plant to the stem and the leaves. Therefore, the direction of travel is in just one way, upwards only. No energy is needed as this is a physical process. The phloem transports dissolved food, i.e. glucose, which is made in the process of photosynthesis in the leaves. This needs to be transported up through the phloem to the developing tissue or the meristem, and down the phloem to the roots. The roots can't carry out photosynthesis, so they need to get their glucose that comes from the leaves. Therefore, the direction of travel is up and down the plant. And finally, yes, energy is needed for this process, and this is supplied by the companion cells adjacent to the phloem cells. Just to note, you do need to be able to link structure to function, which is covered in the specialized cells video of topic 1. Another note is that the meristem tissue is another tissue you need to know. This is found in the shoot and the roots, and again, hopefully you've already covered this in topic 1, stem cells. 15. Transpiration. Plants require carbon dioxide for photosynthesis. This comes from the air around them and enters the leaves through small pores called stomata. These can be open, like this one which shows the stoma, which is a singular of stomata, or they can be closed so you can't see it. This opening and closing is controlled by the guard cells, which are found on either side of the stomata here. In this diagram, the guard cells are turgid. That means they're full of water, and this gives them this particular shape that causes the stoma to open. When the guard cells lose water, they become flaccid, and their shape becomes more rectangular, which closes the stoma. The guard cells control gas exchange. When the stomata are open, carbon dioxide diffuses into the leaf, where it's then used for photosynthesis. Oxygen is produced in this process, and it will then diffuse out of the leaf, at least the excess that isn't needed for respiration. Unfortunately, water vapor is also able to escape through these open pores. Let's take a look at this in more detail. Water evaporates from the surface of cells in the air spaces of the spongy mesophyll. When it evaporates, it turns into water vapor, and this water vapor then leaves through the stomata. This process of water loss through the stomata is called transpiration. That's a key word to learn. Now let's look at the effect it has. The orange arrows show the path of water through the plant. Water is getting pulled up through the xylem, through the stem and the leaves, to replace the lost water. So more water will need to be taken up by the roots in the process of osmosis. This movement of water through the whole plant is referred to as the transpiration stream. It's important to remember that transpiration is driven by the evaporation of water in the leaves. 16. Factors affecting the rate of transpiration. We learned in the previous video that stomata allow gas exchange to take place, but they also allow water vapor to escape. This is called transpiration, and certain factors affect the rate of transpiration. Because the stomata open to allow carbon dioxide to diffuse in for photosynthesis, conditions that affect the rate of photosynthesis will also affect the rate of transpiration. These factors include light intensity and temperature. You'll learn how these affect the rate of photosynthesis in the bioenergetics topic, but for now, you need to know that increasing each of these increases the rate of photosynthesis up to a point, which therefore increases the rate of transpiration. Temperature also gives the water vapor molecules more energy, so they're able to move around more rapidly, resulting in faster diffusion. And when there's faster diffusion, there's a faster rate of transpiration. Conditions that affect evaporation also affect the rate of transpiration, as it's the evaporation of water from the cells inside the leaf that results in transpiration. Humidity is one of these factors. This is how much water vapor is in the air. The air on the left is very humid, as there's lots of water molecules. This means there's a lower concentration gradient between the leaves and the outside air, so diffusion and therefore the rate of transpiration will happen more slowly. If the air is really dry, like you see here, then there's going to be a steeper concentration gradient between the leaves and the outside air, so diffusion and transpiration occur more rapidly. Finally, we've got air movement, or how windy it is. Take a look at what the wind does here. It moves the water molecules quickly away from the surface of the leaf. That leaves a higher concentration inside the leaf than outside the leaf. So we can say that lots of air movement, or wind, maintains a steep concentration gradient, which means a faster rate of evaporation and therefore a higher rate of transpiration as well. It's important to remember these 4 factors that are affecting the rate of transpiration. Plants want to reduce water loss while still carrying out as much photosynthesis as possible. This is especially important for plants living in hot, dry, windy, and bright environments. One adaptation they have to do this is the waxy cuticle. This waterproofs the leaf, preventing water loss. It can be very thick depending on the plant's environment. Secondly, they can wilt. This means that the leaves drip down. This is protective as it reduces the surface area for transpiration to occur. It also means that less direct sunlight is absorbed by the leaves. And finally, an extreme solution is to close the stomata. This of course prevents water loss, but it also prevents photosynthesis. Without gas exchange, no carbon dioxide can diffuse in, and therefore no photosynthesis can occur. The plant will reopen them when the conditions are more favourable. Okay, so it's really likely that you're going to get a math skill question when you're talking about the rate of transpiration. So let's take a look at this piece of apparatus that's used to calculate the rate of transpiration, and then we'll do the questions to practice this skill. So this apparatus is called a potometer. At one end you've got a chute, and you're going to introduce an air bubble into the capillary tube. You just do this by taking the pedometer out of the water and blotting it with some paper towel. Then over here we have a ruler that's going to measure how far the air bubble moves, and we've got a reservoir of water to flush out the air bubble and start again. As transpiration occurs, water is drawn up and this pulls the air bubble along with it. The rate of transpiration can then be calculated by how quickly the air bubble moves. When we say quickly, we mean the speed, which is calculated with distance divided by time. Okay, now we've understood how to use the apparatus, let's try these questions. Calculate the rate of transpiration for this chute under A, normal conditions, and B, windy conditions. Okay, so for A, the air bubble moves 5 millimeters in 8 minutes. So we're going to do speed equals distance divided by time. And the distance is 5 millimeters, and the time is 8 minutes. On our calculator, this comes out to 0.625 millimeters per minute. Now let's look at B. This is under windy conditions. How do you think you would create these conditions in the lab? Well, you could simply use a fan. This can generate some wind speed. Why not pause the video and then try this question yourself first? Ready? So again, we're going to do speed equals distance, which here is 12 millimeters, divided by time, which in this case is 9 minutes. When we do this on the calculator, we get 1.33 recurring millimeters per minute. Now, it's more than possible that you will get a question that asks you to compare the different rates of transpiration. So be prepared to say which one is faster and give a reason why. Our third topic is infection and response, and we're starting with number 1, communicable diseases. Communicable diseases are ones that can be transmitted from one organism to another, i.e., they are infectious. Pathogens are microorganisms that cause infectious diseases. These include microorganisms from 4 groups. We've got bacteria, viruses, protists, and fungi. You'll learn specific diseases from each group later in this topic. Pathogens can infect plants or animals, and they spread by 3 different methods. The first is direct contact. This is when the infected individual directly touches a healthy individual, and this passes the disease on. This is very common in plant diseases, but can also happen in humans with things like STDs. Secondly, we've got water. Some pathogens can spread in the water and infect the individual when they drink the contaminated water. It enters the body through the digestive system. And thirdly, we've got the air. Many diseases can spread in the air, including droplet infection. When you have a cold and sneeze, millions of droplets containing pathogens are spread into the air, and other people may breathe them in. The majority of communicable diseases are caused by bacteria and viruses. Let's look at these 2 groups of pathogens and how they differ. Bacteria are larger than viruses, and there are many good bacteria that play an important role in our digestive system. It's actually estimated that the average human has roughly the same number of bacterial cells as human cells in their body. It's no surprise then that bacteria can reproduce rapidly in the process of binary fission. They can produce poisons called toxins, and these damage the tissues of the body. This is what actually makes us feel ill. Viruses then are a lot smaller than bacteria. They're teeny tiny. And these actually live inside the body cells of the host. Once there, they also reproduce rapidly, but they require the host's body cell to actually do this. They damage and eventually destroy the host cells. This happens when they've reproduced enough times, they burst out of the cell and infect more cells. There are 4 main ways to reduce the spread of disease. The first is simple hygiene measures. These include things like washing your hands after using the bathroom, using disinfectants to clean kitchen and bathroom surfaces, and food safety things like keeping raw meat separate from other foods, and using tissues when sneezing or coughing. This minimizes pathogen spreading in the air. Secondly is destroying vectors. Vectors are organisms that carry disease. For example, Mosquitoes carry the protist that causes malaria. If the mosquito is destroyed, then the disease cannot spread. We can also isolate infected individuals until they're better. This is done when the disease is highly infectious or dangerous, for example, Ebola. Isolating them reduces the number of healthy people who come into contact with them. And finally, we've got vaccinations. You'll learn a lot more about this in the vaccinations video. But this prevents disease by introducing a small amount of a dead or harmless pathogen into the body. The immune system then learns how to destroy the pathogen and can do this if it ever comes into contact with the full disease. 2. Viral diseases. Viruses live and reproduce inside host body cells. This makes treating viral diseases very hard, as to destroy the viruses, you have to damage and destroy your own body cells. Instead, the focus is on preventing the spread of viral diseases. So we'll look at how this is done for each of the 3 diseases. First up is measles. The measles virus is spread by droplet infection in the air. This happens when an infected person sneezes or coughs, and another person inhales the virus. Symptoms of this disease include fever and a red skin rash that covers the whole body. It's a serious illness, and it can even be fatal if complications arise. Because of this, infected individuals are advised to be isolated, and most young children are vaccinated against measles. HIV is spread by sexual contact or by the exchange of body fluids such as blood. This can happen when drug users share needles. You can see in this picture here that there's some residual blood on the needle. HIV initially causes flu-like symptoms. If it's caught early enough, antiretroviral drugs can suppress the virus, but they don't cure it. If it's not caught early, then it can develop into late-stage HIV or AIDS. This is when the body's immune system becomes very badly damaged, and it means it can no longer deal with other infections or cancers. Patients can ultimately die from something like the common cold, as their immune system cannot fight it off. Because of this, people with AIDS have a much lower life expectancy. To prevent the spread of HIV, blood transfusions should be screened for this disease. Needles should never be shared. And condoms should be used before any sexual contact is made, including oral sex. Mothers can also be screened for HIV during pregnancy. If found to be positive, they'll receive specialist care and should not breastfeed their babies. Tobacco mosaic virus, or TMV, affects many species of plants, including tobacco plants and tomatoes. It's spread by direct contact and vectors, which are other organisms that can carry the disease pathogen. This means that the disease can spread easily and quickly. TMV damages leaves by giving them a distinct mosaic pattern of discoloration. You can see this here. This reduces photosynthesis, so the plant growth is really restricted, and therefore farmers will get a smaller yield for their crops. To reduce the spread, farmers can use some form of pest control, and they may grow TMV-resistant crops. 3. Bacterial diseases. Salmonella is a common form of food poisoning. It's a bacterium found in the guts of many different animals, and it can also be found in eggs and raw meat. It spreads when undercooked meat is eaten, or when food has been prepared in unhygienic conditions. For example, if a knife or chopping board were used for raw meat, and then used to prepare a salad without being washed, the salad would get contaminated with the Salmonella bacteria. This is spreading by direct contact. The symptoms of Salmonella food poisoning include fever, vomiting, abdominal cramps, and diarrhea. Not very nice at all. To prevent this from happening, in the UK, poultry are vaccinated against Salmonella to control the spread. Practicing safe food hygiene is also important. Cooking meat thoroughly, keeping raw meat separate from other foods, and washing hands and utensils properly after contact with raw meat. This means using warm water and soap. Gonorrhea is a sexually transmitted disease, also known as an STD. This means the bacteria spread when there is any sexual contact between people. This includes unprotected vaginal, oral, and anal sex. The symptoms are a thick yellow or green discharge from the vagina or penis, and there's pain upon urinating, which just means peeing. And this isn't on the specification, but around 1 in 10 infected men and almost half of infected women do not experience any symptoms. So a lack of symptoms doesn't necessarily mean that a person is disease-free. Gonorrhea can be treated with antibiotics like penicillin. However, many antibiotic-resistant strains have since emerged, so it's becoming a lot more difficult to treat. The spread of gonorrhea can be prevented by using condoms, dental dams, etc. during sexual contact. 4. Fungal and protist diseases. Fungal diseases aren't that common in humans, with the exception of athlete's foot. They're much more common in plants. The one you need to know is rose black spot. This affects the leaves of roses. The spores of this fungus spread in the environment by wind and by water. The symptoms of rose black spot are in the name. There are purple or black spots developing on plant leaves. The leaves often turn yellow and can drop off the plant earlier than normal. Both of these symptoms result in less photosynthesis. Having purple or black spots means there's less chlorophyll in the leaf, and having less leaves or yellow leaves also reduces the amount of chlorophyll. This results in less growth of the plant, and can even mean that there are less or smaller roses. This disease can be treated in 2 ways. Firstly, we've got chemical fungicides that kill the spores, and secondly is removing and destroying any affected leaves. This is usually done by burning them. Malaria is a deadly disease caused by protists. The malarial protist has a life cycle that involves the mosquito. Mosquitoes are vectors for malaria, meaning that they carry the disease. Only female mosquitoes bite humans, and if the mosquito is infected, it passes the protist into the human's bloodstream. Once there, it circulates around the body and damages both the blood cells and the liver cells. The symptoms of malaria are recurrent episodes of fever and shaking. It can be fatal if it isn't treated promptly. In many countries where malaria is prevalent, like sub-Saharan Africa, the medicine isn't easily accessible or it's very expensive, so unfortunately, many people die from this disease. The spread of malaria is controlled by dealing with the vectors, i.e. dealing with mosquitoes. First up is preventing mosquitoes from biting humans. This can be done using mosquito nets and insecticide. Secondly is preventing mosquitoes from breeding. They require standing water to breed, which is just still water. So removing any standing water from areas around the home can prevent them breeding near humans. This reduces their population. 5. Human defense systems. The first line of defense is non-specific, meaning it's used against all types of pathogens. There are 4 parts to this. First up is the nose. Your nose contains hairs and mucus, which can trap any pathogens that enter through it. Then we've got the skin. You're probably aware that the skin acts as a barrier, preventing pathogens from entering the body. If it gets cut, the body repairs it by forming a scab and new skin. But the skin does more than just this. It also produces antimicrobial secretions that can destroy some bacteria. Healthy skin is also covered with a layer of microorganisms that act as an extra barrier to the entry of pathogens. Over here, we've got the trachea and bronchi, which we learnt about in the lungs and gas exchange video. Both of these tubes produce mucus to trap pathogens. They also have lots of cilia. Cilia are tiny hairs which line all the way down the tubes. These can waft the mucus back up to the throat, where it can then be swallowed. When it is swallowed, it goes down the esophagus, which is a big long tube, and into the stomach. And the stomach is number 4. This organ contains hydrochloric acid, which is strong enough to kill most pathogens that enter it, These come from the mucus and from food and drinks. If any pathogens get past the first line of defense, they will meet the immune system. The immune system is made up of different white blood cells, and they can do 3 things to defend against pathogens. First up is phagocytosis. This is when a white blood cell engulfs, which means it sort of surrounds and absorbs. It engulfs the pathogen and then digests it, which breaks it down and ultimately destroys it. Next up is antibody production. Pathogens have antigens on their surface. You can see some of these over here. These alert the immune system to the fact that the pathogen is foreign to the body. Some white blood cells can make antibodies. These are special proteins, and they have a specific shape that means that they can attach to the antigen. When they attach to it, they label it for destruction. There's a unique antibody for each type of pathogen. And lastly, we've got antitoxin production. Antitoxins are able to counteract, or in other words, neutralize, the toxins released by the pathogens. And this means they can't harm you or make you feel ill. Over here, you can see them locking together. 6. Vaccinations. In the previous video, we learned about how the immune system defends the body against infection. The issue is that some diseases act quickly before the white blood cells have time to make the right antibodies. Some of these are deadly diseases like meningitis. But thanks to Edward Jenner and other scientists, we have vaccinations to prepare the immune system. Here's how they work. Number 1. A small quantity of dead or inactive pathogen is put into the body. This is called a vaccine, and it's normally delivered via an injection. The white blood cells will then detect the antigens of the pathogen and begin producing the correct antibodies. This takes some time, so it's a pretty slow response. But that's okay, because if the real pathogen re-enters the body as the full disease, then the white blood cells can produce the antibodies much more rapidly. This time, it's a fast response. The pathogen will be destroyed quickly before they can cause any symptoms of illness. Vaccinations have saved millions of lives around the world. If a large enough proportion of a population is immune to a communicable disease, then the disease will not be able to spread easily and may even be eradicated. Over here in this picture, you can see that this pathogen is struggling to spread because if you look at the members of the population, they've all been vaccinated. This only works if it's many people in the population. But if there are a very small number of people who haven't been vaccinated, for example, if they're very young or very old or have an illness, then they'll still gain immunity, as the disease cannot spread to them from the other people. This is known as herd immunity. That's an important phrase to remember, so let's highlight it. And it's also important to remember that this only works if there's a very small number of people who aren't vaccinated. In this example here, let's say that this lady hasn't been vaccinated because she has an illness that prevents her from doing so. She'll still be protected from catching this pathogen if everybody around her is vaccinated. The World Health Organization is aiming to establish global herd immunity for a number of diseases. Ongoing education and financial support is necessary to make this possible. 7. Antibiotics and painkillers. Antibiotics are widely used in medicine to cure bacterial diseases and have greatly reduced the number of deaths from these pathogens. They're normally taken orally, which means they're swallowed through the mouth, as pills or syrup. Once they've entered the body, they kill the infectious bacteria while leaving your body cells unharmed. One example of an antibiotic is penicillin. You need to learn this example, so we'll highlight it. Specific antibiotics work against specific bacteria, so it's important to use the right antibiotic to cure each disease. For example, this amoxicillin syrup would be able to work against this little bacteria here, but that doesn't mean it'll work against this other species of bacteria. Some strains of bacteria have developed antibiotic resistance, meaning that the antibiotics aren't effective at killing them. This is a real concern, as it takes many years and huge sums of money to develop new antibiotics. Many deaths could occur from antibiotic-resistant bacteria. You'll learn more about this, including how it could be prevented, in the antibiotic resistance video, which is paper 2 content. Antibiotics cannot kill viruses, they only work on bacteria. Viruses reproduce inside your body's cells. Because of this, it's hard to develop drugs that destroy viruses without also damaging the body's tissues. The symptoms of disease can be treated with painkillers and other medicines. These don't actually kill the pathogen, so they don't cure the disease, but they do help you feel a wee bit better while your immune system actually overcomes it. For example, this girl here has a viral infection that's causing her to have a headache. She can take painkillers for this to help her relieve her symptoms while her immune system destroys the viruses. 8. The discovery and development of drugs. Drugs are used in medicine to cure diseases and treat symptoms. Traditionally, drugs were extracted from plants and microorganisms. There are 3 examples of this to learn. The heart drug digitalis originates from foxgloves. This plant is pretty common in the UK, but it's poisonous if eaten directly. The painkiller aspirin originates from willow trees, which look like this. And the antibiotic penicillin, which you learned about in the previous video, originates from the penicillin mold. Alexander Fleming discovered this accidentally when he left some bacterial cultures open for a while. He later found that those that had mold growing on them had clear rings around the mold where the bacteria had been killed. He struggled to extract the penicillin from the mold, but luckily 2 other scientists called Florey and Chien followed up on his work and were able to extract the world's first antibiotic. Nowadays, most new drugs are synthesized by chemists in the pharmaceutical industry. However, the starting point may still be a chemical extracted from a plant. This is one reason why maintaining biodiversity is so important. New medical drugs have to be tested and trialled before being used to check 3 things. First is toxicity. This means checking if it's poisonous or not. Secondly is efficacy, which means it has the intended effect, i.e., it treats the disease. And thirdly is the dosage. This is checking the amount of drug that needs to be taken to work best. So I have a tip for you. You can remember these 3 with the word TED. This is just the first letter of each of the words, and you can make the link that teddies need to be tested too to make sure they're safe before they're sold to children. See if it helps. Okay, the first stage of testing is preclinical testing. This is testing the drug in a laboratory using cells, tissues, sometimes whole organs, and live animals. Cells and tissues are used first before testing on live animals. There are a lot of strict rules before testing can be done on real animals. If the drug passes the test for the preclinical trials, then clinical trials may begin. These use healthy volunteers and patients. So first up, very low doses are given to healthy volunteers to check if they're safe or if there are any side effects. If they are safe, then they're tested on a small number of patients. This is to see if it actually treats the disease. And if it does, then larger further trials are carried out to find the optimum dose for the drug. Clinical trials are double-blind trials. This means that some patients are given a placebo, while other patients are given the real drug. The placebo and the real drug look identical, and they're delivered in the same way, i.e. both as pills or both as injections. Neither the doctor nor the patient know what they've been given. This is what makes it a double-blind trial. Only the researcher knows, and this is done to check if the drug really works or if any improvements in health are just due to the placebo effect. The results of drugs testing and trials are only published after scrutiny by peer review. This is when other scientists check the results to make sure they're robust and true. If they are, they'll get published in a scientific journal, and then national health bodies will look at the results and decide which drugs should be produced and offered on the NHS. 9. Monoclonal antibodies. In a previous video, we covered how antibodies work. These are proteins made by white blood cells that can attach to antigens. Antigens are proteins often found on the surfaces of cells. Today, we're looking at how large amounts of antibodies can be made in the lab. These are monoclonal antibodies. In the first stage, a mouse is stimulated so that its white blood cells make antibodies for a particular antigen. As you're studying biology only or triple, you need to learn that these white blood cells are called lymphocytes. These lymphocytes are then combined with a particular type of tumour cell. This makes a new cell called a hybridoma. Hybridoma cells have the properties of both cell types. It can produce antibodies like a lymphocyte, and it can divide rapidly like a tumour cell. Single hybridoma cells are then cloned to produce many identical cells. These can all divide rapidly to produce the same antibody. A large amount of the monoclonal antibodies can then be collected and purified for use. The uses of monoclonal antibodies are all possible because the antibodies will only bind to one specific type of antigen, which allows it to target specific cells in the body or chemicals like hormones. You need to be able to describe some of the uses of monoclonal antibodies, and you can also expect an application question with this topic to explain how the monoclonal antibodies work. There are 4 to learn. The first is pregnancy tests. A specific hormone is made during the early stages of pregnancy, and if it's present, monoclonal antibodies, or MABs, will bind to it and cause a colour change to indicate pregnancy, like this. They can also be bound to a fluorescent dye, and they're then used in research to locate or identify specific molecules in a cell or the whole cells themselves. Monoclonal antibodies can also be used to detect diseases. This can be done by binding to the pathogen's antigens, like this. They can also bind to other chemicals or hormones in the blood that are only produced in high levels if a person has a disease. And finally, they can be used to treat some diseases, like cancer. In this case, a substance such as a toxic drug, a radioactive substance, or a chemical that stops the growth of the cells can all be attached to the monoclonal antibody. When it then binds to the tumour cell's antigens, it delivers the substance to the cell. Monoclonal antibodies deliver substances to disease cells without damaging the body's healthy cells. This is a huge advantage of monoclonal antibodies. Conventional drugs are carried all over the body and are much harder to target to specific cells or tissues, resulting in damage to healthy cells. Targeting cells based on their antigens allows a much more specific response. There is one big disadvantage to monoclonal antibodies. Because they were made using mouse lymphocytes, monoclonal antibodies have created more side effects than expected. Because of this, they're not yet as widely used as hoped when they were first developed. Researchers are continuing to improve the production of monoclonal antibodies to try and reduce the side effects, and doctors are also able to anticipate and treat side effects, so it's hoped that they will be more widely used in the future. 10. Plant diseases. Earlier in the infection and response topic, you learned about plant diseases including TMV and rose black spot. Viral, bacterial, and fungal pathogens can infect plants, but there are other ways that plants can be damaged. The first is by insects, in particular aphids. Aphids are small green insects that have a sharp mouthpiece. They insert this into the plant stem and penetrate the phloem vessel, sucking the sap out of it. This deprives the plant of the sugar it made during photosynthesis, which damages the plant and reduces its growth. Aphids can also be vectors for disease. This means they carry the pathogens to the plant. Gardeners can destroy aphids by using chemical pesticides or biological pesticides. This is when predators like ladybirds are released into the area the plants are grown in. Here, they eat the aphids, removing the pest. Ion deficiency can also damage plants. This is when the soil doesn't have enough of certain ions, and there are two you need to know. Nitrate ions are needed for protein synthesis and therefore growth, so nitrate ion deficiency causes stunted growth, i.e. the plant is smaller. Magnesium ions are used to make chlorophyll. When there isn't enough magnesium, less chlorophyll is made and the leaves look pale or yellow coloured. There is reduced growth as a result. This condition is called chlorosis. Knowing about the ions needed for plants to grow at their best allows gardeners and horticulturists to provide the optimum conditions for plants by enriching the soil with all of the ions that they need. This next part is for higher tier only, so if you're studying foundation, just skip ahead to the questions. Okay, you need to know how plant diseases can be detected. Pause the video and see how many you can spot in the picture below. Alright, there are 7 indicators of disease. Let's see how many you spotted. Here we've got discoloration, presence of pests, spots on leaves like rose black spot, areas of decay or rot, malformed leaves or stems, and here we've got abnormal growth, and finally stunted growth. So there are 7 to memorize. Once you've spotted an indicator of a disease, the next step is to identify which disease is causing it. This can be tricky as many plant diseases have overlapping symptoms. There are 3 main ways to identify disease. The first is using a gardening manual or website. You can compare the diseased plant with descriptions and pictures of each disease. Secondly is taking infected plants or samples to a laboratory to identify the pathogens using something like microscopy or DNA analysis. This may be done for crop plants or forests when the disease affects many plants. And finally, we can use testing kits that contain monoclonal antibodies. These will bind to antigens on certain pathogens if they are present. You learned about this in the previous video. Once the disease has been identified, appropriate action to treat or contain it can happen. And finally, 11. Plant defenses. Plant defenses can be split into 3 groups: physical, chemical, and mechanical. Let's start with physical defenses. Plants have a cellulose cell wall that makes it harder for pathogens to enter the cells. The leaves can have a tough waxy cuticle that acts as an additional barrier to pathogens. Trees have a thick bark, and plants without this have layers of dead cells around the stem. This is an extra layer of protection, and it helps remove pathogens when the dead cells are shed or the bark falls off. Now let's look at chemical defenses. Many plants produce antimicrobial chemicals that can protect them against pathogens. In the Developing Drugs video, we saw that chemicals from plants form the basis of many traditional medicines based on this property. Some plants produce poisons, for example foxgloves and nettles. This deters herbivores from eating them. Herbivores include large animals like cows and smaller animals like insects. If the animal feels unwell or ill after eating the plant, then it's unlikely to eat the same type of plant again. Now we'll look at mechanical defenses. Hairs, like the ones you see on nettle leaves, and thorns, like the ones you see on rose plant stems, deter animals from eating the plant by hurting them. Additionally, insects find it harder to lay eggs on plants if they've got hairs on the leaves. Mimicry is something that's used to trick animals. The plant copies something to make it appear off-putting. For example, the passionflower vine has evolved to have small yellow spots on its leaves. These look like butterfly eggs. This deters butterflies from laying eggs on the leaves, as it appears there will be more competition from other caterpillars when the eggs hatch. The butterflies will lay their eggs on different plant leaves, reducing damage to the passionflower vine. And finally, Leaves that can drip or curl when touched can scare animals away and also cause insects to fall off or fly away from the plant. Our fourth and final topic is bioenergetics, and we'll be kicking it off with number 1, photosynthesis. Photosynthesis is a fundamental process that has already come up in lots of earlier topics like cell biology. It's how plants harness the sun's energy to make food. And this happens in the green parts of the plant, like the leaves and the stem, in the chloroplasts, these little green bits here. This is the word equation. Carbon dioxide plus water, arrow, and light can go along the arrow, glucose plus oxygen. We put light along the arrow because it's not a chemical, but it is a condition needed for photosynthesis to happen. If you're given a word full with just 2 spaces, make sure you put in the chemicals and not light. Okay, let's look at where each of the reactants comes from. CO2 comes from the air around the leaf and diffuses in through the stomata, and water comes from the soil and is absorbed by osmosis. You also need to know the chemical symbols for each of these substances. Carbon dioxide is just CO2, water is H2O, light we don't need to know because it's not a chemical, glucose is C6H12O6, and oxygen is O2. So make sure you have a go at memorizing those. Now, it isn't on the spec, but if you want to practice some chemistry skills, pause the video and try and balance this equation. We just put a 6 in front of everything that isn't glucose. Now, because light from the environment needs to be taken into the chloroplasts for photosynthesis to occur, we can describe it as an endothermic reaction. That's an important phrase to learn. It's endothermic because N means in, and light needs to be taken into the chloroplasts. And just a reminder that the chloroplasts contain chlorophyll, the green pigment, that's what's actually absorbing the light. Leaves are adapted to maximize the amount of photosynthesis that can take place. Pause the video and see how many adaptations you can spot in these 2 diagrams. We'll start with their shape. You can see that it gives them a large surface area to absorb more light. They're thin to allow gases to diffuse in and out easily, and they have veins, which contain the xylem to transport water and the phloem to transport glucose. It's important that glucose can move down the plant as well to the roots, as they can't make their own glucose. They also have lots of guard cells on the underside of the leaf, and these regulate gas exchange. The spongy mesophyll has air spaces, and again, these allow gases to diffuse easily. And finally, the palisade mesophyll tissue has many chloroplasts to maximize photosynthesis. Number 2, the rate of photosynthesis. One factor that can affect the rate of photosynthesis is light intensity. In the previous video, we learned that photosynthesis is an endothermic reaction, so light is needed to transfer energy to the chloroplasts for photosynthesis to occur. It makes sense then that the higher the light intensity, the faster the rate of photosynthesis. This doesn't increase at a steady rate, so the line is a curve. Because the rate increases as we increase the light intensity, it means that light was limiting at the lower intensities. So we call light a limiting factor at these points. That's an important phrase to learn, and all of the other factors we're going to look at are also limiting factors. At a certain point, the rate of photosynthesis plateaus, which means it stops increasing. No matter how much brighter it becomes, the rate doesn't increase, because light intensity isn't limiting it anymore. Another limiting factor has been reached. This means that something else is preventing the rate of photosynthesis from increasing. This could be any one of the 3 remaining factors that we're going to look at. For example, carbon dioxide concentration. Carbon dioxide is a reactant in photosynthesis, so as we increase the concentration of it, the rate of photosynthesis will also increase. This can be shown on the graph like so. At the low concentrations of carbon dioxide, this is limiting. So we say the CO2 concentration is a limiting factor at these concentrations. Beyond a certain concentration, carbon dioxide stops being the limiting factor, and instead it's a different factor that limits the rate of photosynthesis. The third factor that can affect the rate of photosynthesis is temperature. All chemical reactions are affected by temperature. Increasing the temperature increases the rate of photosynthesis until it reaches the optimum temperature. After this point, the enzymes that catalyze this reaction become denatured. You can learn more about this in the Factors Affecting Enzymes video. The final limiting factor is the amount of chlorophyll in the leaves of the plant. Chlorophyll is the green pigment found in the chloroplasts that can absorb light for photosynthesis. Reduced chlorophyll means that less photosynthesis can take place and the rate is reduced. This can happen in variegated leaves like the spider plant, which have got white stripes and therefore reduced chlorophyll. It can also happen when a plant is deficient in magnesium ions, and this causes chlorosis. Graphs aren't normally used for this, as chlorophyll isn't a variable that can be easily changed or measured externally, but it would follow a similar shape to the light and carbon dioxide graphs. This next bit is higher tier content only. If you're studying foundation, skip to the quick questions at the end. So the first thing to note is that limiting factors can and do interact with each other. It's important to be able to identify which factor is limiting the rate of photosynthesis. For example, if you were making a cake and had 100 eggs but only 200 grams of flour, then the flour is limiting the size of the cake and is our limiting factor. It works the same way for photosynthesis. If a plant receives a high light intensity but has a low concentration of carbon dioxide, then overall the rate of photosynthesis will be low. This is because the carbon dioxide concentration has limited it. We can show multiple limiting factors on the same graph like this. In this graph, we've got light intensity and carbon dioxide concentration. Temperature was controlled, which means it was kept the same, for example, at 20 degrees Celsius. So let's take a look at our graph. At this part of the graph, light intensity is the limiting factor, because as it increases, the rate of photosynthesis also increases. Let's take a look at the next part of the graph. What do you think is the limiting factor here? Pause and see if you can work it out. The answer is that carbon dioxide is the limiting factor. Here's how we know this. The blue line is 0.04% carbon dioxide. When the carbon dioxide concentration increases to 0.08, the rate of photosynthesis increases. That means that the lower concentration of carbon dioxide was the limiting factor. Now let's look up here. When it's 0.08% of carbon dioxide, what's the limiting factor here? Pause and have a think. Well, the truth is we can't work it out unless we do another experiment. Let's say, for example, that we increase the carbon dioxide concentration again, and this time we change it to 0.12% carbon dioxide. You can see that the lines on the graph are pretty much the same. That means that increasing the carbon dioxide concentration had no effect on the rate of photosynthesis, so the limiting factor is not carbon dioxide concentration. Therefore, it must either be temperature or chlorophyll. You could do another experiment to find out which. As I mentioned, chlorophyll is hard to change externally, so instead we'll change the temperature. And here we are. You can see that the early investigations were done at 20 degrees Celsius, but our final investigation has been increased to 30 degrees Celsius. We can compare this result, but only to the investigation that had the same concentration of CO2. Otherwise, we have two different independent variables. So, we can actually see that when we compare these, the rate of photosynthesis does increase when we increase the temperature. Therefore, temperature was the limiting factor in the earlier investigation. Now I know these graphs may look hard, but with practice, I promise they'll become easier. And just one thing to point out again is that you cannot compare results with two different independent variables. So in this situation, we were only able to compare the two highlighted results because they had the same CO2 concentration. 3. Making the most of photosynthesis. We learned about limiting factors in the previous video and how they can affect the rate of photosynthesis. Farmers and gardeners can remove limiting factors to maximize the rate of photosynthesis. This is often done in a greenhouse, which looks like this. Pause the video and see if you can spot some ways in which this greenhouse increases the rate of photosynthesis. Well, first of all, greenhouses naturally trap heat from the sun, which increases the temperature, and warm temperatures cause a higher rate of photosynthesis. If it gets too hot, greenhouses can be ventilated using open windows. This lets wind in and cools the greenhouse down. In very cold conditions, they can use heaters to provide warmth, ensuring that the optimum temperature for photosynthesis is maintained. Gardeners can also provide higher CO2 concentrations within greenhouses. This is commonly done using paraffin heaters. These release carbon dioxide as they burn and also provide heat. The higher CO2 concentration increases the rate of photosynthesis. Photosynthesis doesn't happen without light, so in natural conditions, plants only photosynthesize during daylight. Artificial lights allow farmers and gardeners to extend the length of time that plants can photosynthesize by turning on lights after the sun has gone down or when it is cloudy. All of these methods of increasing the rate of photosynthesis cost money. The gardeners and farmers need to be sure that the profits from the extra yield of their crops exceeds the costs. Here's one example. This graph shows the yield of tomatoes as the plants receive increasing hours of artificial light. Just looking at this graph, you would say that 10 hours of artificial light is the best, because 10 hours gives the highest yield in tomatoes. But this doesn't take into account the costs. We know that artificial lights use electricity and therefore cost the gardener or farmer money. We would probably therefore change our answer and choose the point in the graph where the increase in yield really slows down. This happens at about 6 hours of artificial light. So this is actually going to be a better number of hours for the farmer because it will increase their profits. Now let's say we had access to the information in this graph. This graph looks at the actual profit. We can see that the profit peaks at 4 hours of artificial light, a different answer than what we came up with previously. But when we look at the previous graph, we can see that between 3 and 4 hours of artificial light was the steepest increase in yield. So it makes sense that this would give the most profit for the farmer. Data analysis questions are really common in this topic, so make sure you practice interpreting graphs and tables. I've got videos for this, and my exam-style questions in my bioenergetics workbook will also focus on this skill. 4. How plants use glucose. Plants make glucose in the process of photosynthesis. While we often say this is food for the plant, in reality, this simple sugar has got many uses. It can be tricky to remember them all, so I use the made-up word phosphorus. This doesn't mean anything, but somehow it sticks in my mind to help me remember the uses when I'm teaching it. Here's what each letter stands for. F and O are fats and oils. Glucose is used to produce these for storage in the plant. They are energy stores if the plant needs it. S is for starch. Glucose can be converted into starch, which is another storage material. This can be converted back into glucose when the plant requires it, for example, at night when it isn't photosynthesizing. R is for respiration, and in this process, glucose is broken down to release energy for the plant. You'll learn more about this in the next video. A and A are for amino acids. Glucose is used to make amino acids, along with nitrate ions that plants get from the soil. The amino acids are then used in protein synthesis, which just means making proteins. And finally, C is for cellulose. Glucose is used to produce cellulose, which is a molecule that strengthens the cell walls of plants, which is this part here. And that is pretty much it. If you don't like the word phosphorus or it doesn't stick in your head, then try coming up with your own mnemonic. This is a brilliant way to remember lots of linked content like this. And if you're using my bioenergetics workbook, I've provided an extra page for you to have a go at making up your mnemonic and an explanation of how to do this. 5. Respiration. Respiration is an exothermic reaction, which means energy is transferred to the environment, i.e., it is released. This energy supplies all of the energy needed for living processes in organisms, for example, movement, keeping warm, and carrying out chemical reactions to build larger molecules. Note that the keeping warm aspect largely applies to mammals. If we were asked about how a plant uses the energy transferred in respiration, then this wouldn't apply. Instead, we could mention how active transport in the roots requires energy to bring mineral ions against a concentration gradient. That's a good link across topics. To supply enough energy for all of these processes, respiration happens continuously in cells. Aerobic respiration can occur when there's adequate oxygen present, and this happens in the mitochondria of the cell. Here's the word equation for it. Glucose plus oxygen, arrow, carbon dioxide plus water. This equation for aerobic respiration is actually the reverse of the photosynthesis equation, so this might help you remember it. Photosynthesis was also an endothermic reaction, but respiration is exothermic. That's another important difference. Okay, you also need to know the symbols for each of these chemicals, so pause the video and see how many you can remember. Ready? Glucose is C6H12O6, oxygen is O2, carbon dioxide is CO2, and water is H2O. Now we'll look at anaerobic respiration. This occurs in the absence of oxygen, and it can happen in our bodies when the muscles are working very hard, for example, when we're sprinting. Less energy is transferred than in aerobic respiration because the oxidation of glucose is incomplete. This means that the glucose isn't fully broken down, so less energy is released. Here's the word equation for anaerobic respiration in the muscles of animals. Glucose is broken down to give us lactic acid, and a small amount of energy is released. Anaerobic respiration in plant and yeast cells produces different products. So here's the word equation for it. Glucose breaks down to give carbon dioxide plus ethanol. Anaerobic respiration in yeast cells is called fermentation. That's an important word to learn. And this process has economic importance as it's used in the manufacture of alcoholic drinks, for example, beer. This is possible because ethanol is released, which is a type of alcohol. It's also used to make bread, as the CO2 helps the bread rise, and the ethanol just evaporates as the bread is baked. 6. Response to exercise. Exercise involves the contraction and relaxation of our muscles to move the body. This increases the demand for energy, which is transferred during respiration. As we learned in the previous video, more energy is transferred during aerobic respiration than anaerobic respiration. So our bodies will try to respire aerobically for as long as possible. To help with this, 3 main changes occur. Firstly, our heart rate increases. This means more blood is pumped to the muscles each minute. Secondly, our breathing rate increases. This means we take more breaths per minute, getting more air, and therefore more oxygen into our body. And finally, our breathing volume increases. This means we're breathing more deeply, taking more air into our lungs, resulting in more oxygen entering the bloodstream. The overall result is that our muscles receive more oxygenated blood, so the mitochondria can carry out more aerobic respiration. Long periods of vigorous activity cause muscles to become fatigued and stop contracting efficiently. You've probably experienced this yourself when exercising. Your muscles get sore and tired. One cause of this is lactic acid. This is produced during anaerobic respiration due to insufficient oxygen. And as we saw in the previous video, the incomplete oxidation of glucose causes a buildup of lactic acid. This also causes an oxygen debt. The explanation of oxygen debt is higher tier content, so if you're studying foundation, feel free to skip ahead to the questions. Okay, lactic acid is a waste product that the body needs to remove. It isn't a gas, so it can't just be breathed out like carbon dioxide. Instead, blood flowing through the muscles will pick it up and transport it to the liver. Once there, the lactic acid is converted back into glucose using oxygen. The amount of extra oxygen needed to do this is called the oxygen debt. That's an important phrase to learn. This is the reason why we continue to breathe heavily and quickly after exercise. The body still requires a higher amount of oxygen to repay the oxygen debt and convert the lactic acid back into glucose. And our very last section is number 7, metabolism. I've briefly mentioned metabolism in my enzymes video. If you've seen it, pause the video and see if you can think of the definition for metabolism. Metabolism is the sum of all the reactions in a cell or organism. You can remember this with this little graphic here. These are reactions in which molecules are made or broken down. There are quite a few examples for this across the specification, and in this video we're linking them together. There are 6 to learn. The first is the digestion and synthesis of carbohydrates, lipids, and proteins. Can you remember what each one breaks down into? Pause the video and see. Okay, carbohydrates break down into sugars, lipids break down into glycerol and fatty acids, and proteins break down into amino acids. An extra bit of detail to learn is that lipids are synthesized from 1 glycerol molecule and 3 fatty acid molecules. Synthesis is the opposite of digestion or breaking down, so it's just going in the reverse, and the small molecules build up the bigger molecules. Our next process is that glucose can be converted into starch or cellulose in plant cells. In animal cells, it can be converted into glycogen. This is an energy store for later use. Glucose and nitrate ions are used to form amino acids. These are then used to synthesize proteins, as we saw previously. Proteins are just chains of amino acids. Excess proteins in the body are broken down to form urea for excretion. This is turned into urine in the kidneys, and it then leaves the body as urine or pee. Next up is respiration. This is a metabolic process in which the breakdown of glucose transfers energy to the cell. And finally, we've got photosynthesis. This is also a metabolic process, and it occurs in plants and algae to produce food using light. There are hundreds of thousands of metabolic processes, but these are the examples you need to learn for the exams. If you finished your revision notes or are using my study along workbooks, then I suggest going through them and finding these examples. Then mark the pages with the little M for metabolism to remind you when you next revise it.
I hope you found that video useful and are feeling prepared for your exams. If you did find it helpful, then please hit my icon over there to subscribe or the big red button down below. Thanks again for watching and good luck for your exams.